Welcome to Biowise with Aanchal. Exam knocking at the door, prelims in January, and we have to finish the portion in a very quick manner. So, 6 days, 6 videos, and 6 units, and you'll complete the entire syllabus in biology. So, let us begin without wasting time with the first unit, that is basic biology. In this unit, we are going to study about cell cycle, cell division, structure of chromosomes, genetics, Mendel's law of genetics, X-linked inheritance, and many more important topics that is going to be covered in this video. Now, this basic unit of biology is basically for around 20 to 25 marks in your examination as per the analysis done for the last 3 years paper. So let us begin with the chapter, the cell. Now, cell, the chapter itself is not important for your exam point of view. The basic understanding of the chapter is required to draw certain diagrams that is required in your examination. So just go through this lesson, know about what is the difference between a plant cell and animal cell, unique structures of plant cells and animal cells, what is cell theory, that will be more than enough. Structure of chloroplast, mitochondria is important, right? So let's begin with the important concept of basic biology, the structure of chromosomes. This is the diagram for the chromosomes. Now you must have seen I have used the traditional pink and blue to depict the chromosomes from the mother and the father. Now these set of chromosomes which we inherit from our parents is called homologous chromosomes. A pair of chromosomes of same size, structure, inherited from either of our parents is called homologous chromosomes. Now you have understood the term homologous chromosome, right? Fine. Now when you see the chromosome and you divide it into half, okay, this single wing-like structure is called a chromatid. Okay, so these two are called sister chromatids and these two are called non-sister chromatids. Understood? That is, these are non-sister chromatids, these are sister chromatids. The point of attachment of the chromatids is called a centromere. A centromere. Fine. If you have understood the difference, that chromosome, the entire set is, this entire structure is a chromosome. This single unit is called a chromatid. These two wings of the same chromosome after the division is the sister chromatid. The two wings of the different chromosomes but the homologous ones are called non-sister chromatids. These non-sister chromatids tend to exchange their genetic material during the process of synapsis, right? So you can see here this structure where a cross is formed. It is cross is chiasma. The process of exchange is called synapsis, right? It's called synapsis, chiasma formation, and this is crossing over that is taking place. Which phase it happens? It happens in meiosis. It happens in meiosis, prophase 1. Okay, we'll study about it. So this all things you are going to remember. Synapsis has got cis word that happens in crossing over, exchange of chromatids, segments of the non-sister chromosomes, non-sister chromatids, that is called synapsis. There is one more word, that is synapse, that is in the nervous system, that is junction of neuron for communication, that is in nervous system. Synapsis is cell division where crossing over takes place. That crossing over leads to variation. That is why we all are different, because there is always a recombinant chromosome which is being formed because of the exchange of segments of the non-sister chromatids. Now, based on the chromosomes, there are few types of chromosomes, that is called allosome and autosome. Allosomes are also called sex chromosomes. that determines that what gender will be, either a boy or a girl, and autosome is a general chromosome that both the genders have. So the total number of chromosomes that the human being has is 46, or you can say 23 pairs. Each set we inherit from our parents. That is 23 from the mother, 23 from the father, total makes 46. Okay. Now in this case, for a male, you have 44 plus XY, and for female you have got 44 plus XX. Is that clear? So I hope the concept of chromosome, chromatids, sister chromatids, non-sister chromatids, allosomes, autosomes, homologous chromosomes is understood. So let us proceed with the next part of the lesson, that is cell cycle. This is your cell cycle. Cutting the time short, I have already drawn the diagram. Now you can see here, this is your cell cycle, which has got 4 phases: G1, S, G2 and mitosis, M phase. Okay, G1 phase is also called the cell growth phase. In this, what happens, the cell which has divided by the process of mitosis enters into the G1 phase, where it matures, grow in size, increases the amount of RNA and proteins required for DNA synthesis. At the same time, there are few cells which enter into the G0 phase that they will never be able to divide. They will not divide in their lifetime, like your neurons, like your nephrons. So they stay in the G0 phase, that is, they do not enter into the cell division phase. Then after the cell has grown, matured, it enters into the S phase, where the DNA multiplies, okay, where the DNA becomes double. After that, it enters into the G2 phase, which is also called the second growth phase, where necessary RNA and proteins required for the mitotic phase is needed. Then the cell enters into the mitotic phase, which is the division phase. How you are going to remember the phases order? It is PMAT. What is PMAT? Prophase, metaphase, anaphase, telophase, in which prophase is the longest phase and anaphase is the shortest phase. What happens in the interphase? Now see, this is the cell division phase, a small part. Major part of the cell life is the interphase, where the cell just rests and prepares to divide. So when you have a cell and it is in the interphase stage, you have the nucleus and the chromatin fibers, and you have the centriole. Okay, this is the interphase cell. Then after the interphase, the DNA has doubled, it enters into the prophase. Now in the prophase stage, you will find that the nuclear material starts to disappear, and the chromosome, which was thread like chromatin fibers, it condenses to form the chromosomes. I am drawing it for 4 chromosomes here. You can, the number has to be maintained, children, till the last, okay? And the centrosome, that is the centrioles, start moving towards the opposite pole and the spindle fiber begins. This is your prophase. Okay? Now, after prophase, you have metaphase. Metaphase matlab middle line, in the midway. So here what happens, the nuclear membrane has already disappeared. So the chromosomes will align themselves in the equatorial plate. Okay, they will align themselves in the equatorial plate in the central line. This is how you recognize the cell, in which stage the cell is in. Okay, so you have the centromere attached to the spindle fiber is attached to the centromere. So this is your metaphase. Okay. Now after that, what happens? A for anaphase, A is away. The chromosome starts moving away from each other, and as they move away from each other, the wings should be facing the center line, that is the equatorial plate, because they are pulled apart by the spindle fibers. Okay. So this is how the chromosomes split and move towards the opposite pole. This is your anaphase. And after that, the cell, the the cytokinesis will begin after the cell has got the one set of the chromosomes, that is the diploid set of the chromosomes are received by the daughter cells. Is that clear? And then it will, this entire phase is called karyokinesis, that is, it is the nuclear division. After this, the cell will be separated into two daughter cells. This process of separation of the cytoplasm after the process of karyokinesis is called cytokinesis, that is cell division. The cytoplasm has divided. Karyokinesis means nuclear division. Is that clear? So that any diagram can be given to you in the question, and question can be asked and identify the phase. Identify the phase which precedes the given phase. Precedes matlab the one which is before that. What precedes anaphase? It is metaphase. What is next to anaphase is telophase. There is no late or early that you are going to write in the exam. You have to mention the four phases, whether it is prophase, metaphase, anaphase or telophase. Is that clear? So this is all about your cell cycle and mitosis. Now when I talk about mitosis, mitosis has got few significance. The significance can be remembered as GR. What is GR? G is for growth, R is for repair, R is for replacement, and R is for reproduction. Reproduction means division of the cell. Like, so this is how the significance of mitosis is all about. So this is cell cycle, mitosis, different phases of mitosis. Meiosis is not there in your syllabus. That is, phases are not there in syllabus. What is important for meiosis is significance of meiosis. So significance of meiosis is what? Production of gamete and maintenance of the significant number of the chromosomes in the species. Like 46 chromosome can be maintained in human beings because of meiosis. The cells and meiosis also helps in the formation of gametes, and gametes have half set of chromosomes. That is, after the fusion of the two gametes and the formation of zygote, the original number of the chromosome of the species has to be restored. So question can be asked that if the parent has got these many chromosomes, what is the chromosome number present in the gamete, or what is the chromosome number present in the somatic cells? Okay, please keep in mind that X chromosome is present in all the cells, irrespective of whatever it is, except the sperm cell which has got only Y chromosomes. Okay, so all somatic cells, all male cells, female cells have X chromosomes. Is that clear? Except for the gamete sperm which has got Y chromosome. Fine. So there can be any kind of application based question or competency based question that can be asked on this part of the chapter. We'll have the questions at the last for the practice. So stay tuned till last. If you have not subscribed the channel, you're requested to subscribe it, like, share if you think it is worth it. Now this is all about cell cycle. Let us move to the next part of the chapter, that is DNA and its structure. Now who gave the DNA and its structure was Watson and Crick. Watson and Crick got Nobel Prize in the year 1956 for the structure of DNA, and we have just lost Watson in November 2025 at the age of 97. Crick, he already passed in 2004. So Watson gave the DNA structure which was a double helical structure. Okay, now double helical structure of DNA is given by Watson and Crick, and for that they have got the Nobel Prize in the year 1956. Okay, now according to this structure, DNA, the basic unit of DNA is called a nucleotide. Okay, what is a nucleotide? Nucleotide is you can say a unit of DNA which comprises of sugar, then it has got phosphate group. This is same as in your chemistry wala phosphate. Okay, phosphate group. Then you have got the nitrogenous bases. Okay, nitrogenous bases. Okay, now when you talk about the sugar present in DNA is deoxyribose, and that in RNA is ribose. So what is a ribose sugar? A ribose sugar is a 5-carbon sugar which contains 5 carbon This is how the structure of ribose sugar is all about, and there is OH present here. This is the point of attachment of the nitrogenous base, and here you have the 5 prime carbon which is attached to phosphate. Now this is all numbering system: 1 prime, 2 prime, 3 prime, 4 prime and 5 prime of the sugar. In case of ribose sugar, both these 2 prime and 3 prime position you have OH group. But for deoxy, D matlab absent, oxy is oxygen. So deoxyribose, okay, deoxyribose has got similar structure of the ribose, but in the 2 prime position you have only H, and 3 prime position you have OH. You have 5 prime carbon which is attached to the phosphate. The first prime carbon is attached to the base, and you have 3 prime position where you have addition of new nucleotide will take place. This is about the sugar. Phosphate group is same as what you have in chemistry, that is PO4 3 minus, okay, and nitrogenous base. Now nitrogenous base, if you have to define, these are actually A, T, G and C, and there is one more which is U. Okay. Now T and U, T is present in DNA and U is present in RNA. So T is thymine and U is uracil. Okay. So when you have to see nitrogenous bases, there are two types of nitrogenous bases. One is purine, okay, and the other one is pyrimidine. A and G are the purines, adenine and guanine, and pyrimidine is thymine and cytosine. All these three have got Y in it: pyrimidine, thymine, cytosine, purine, adenine and guanine. You have to remember that A always bonds with T and G always bonds with C. There is 2 hydrogen bond between A and T, and there is 3 hydrogen bond between G and C. Now this can be a platform or a concept for building a question, like if in a DNA double helical structure there are total some number of base pairs. Suppose there are 100 base pairs present in a DNA. Okay, out of these 100 base pairs, if it's a double stranded DNA, 100 base pairs. So now if there are say 20 adenine, then how many cytosine do you have? If it has got 20, there are 100 base pairs. So 100, you can say 20 adenine will base pair with 20 thymine. So 40 is over. 30 guanine will base pair with 30 cytosine. Okay. If they give you, instead of base pairs, they give you there are itna nucleotides. One base pair has got 2 nucleotides. You have to calculate accordingly. Always remember that in one base pair you have got 2 nucleotides. So please see the unit, what type of question is asked. Is that clear? So we can actually calculate the number of A, T, G, C. Even if one number is given, we can calculate the other numbers. So there is one more term, nucleoside. Nucleoside is what? Sugar plus base, no phosphate group. So nucleoside plus phosphate gives you nucleotide. Is that clear? Now there are few other words, like what is a nucleosome? Okay, so nucleosome is the basic unit of a chromosome. Your DNA is too huge. So your single human chromosome has got millions of nucleosomes. So the DNA has to own around the histone proteins, around 8 histone proteins, and they make up a nucleosome. Each nucleosome again forms what to stack themselves one over the other and forms a solenoid, a rod-like structure, which again gets coiled and supercoiled and coiled and then get fit into the chromosomes. Okay, so the basic unit of DNA is nucleotide. Nucleotide will form DNA. DNA will form a nucleosome. Nucleosome will form the chromatin fiber, and chromatin fiber will form the chromosome, arranged in logical sequence. Is that clear? So this is all about the DNA and its structure. Let's move to the next topic, that is genetics. Next is very important, around 10 to 12 marks is asked from genetics only. Okay, now when you talk about genetics, here we have to talk about Mendel. Mendel and his laws of inheritance. That is very important. 100% the question is asked every year that state the law, whether it is law of dominance, law of segregation, or law of independent assortment. All the three laws are very important. Last time it was independent assortment in the paper, but it can be any law in this paper. Okay, so be prepared with the three statements as it is given. Like law of dominance, out of the pair of the contrasting characters, out of the pair of the contrasting characters or traits of the character, the one which is dominant will express itself and the other remains suppressed. This is law of dominance. Law of segregation or law of purity of gametes is out of out of the two contrasting traits, each one separates themselves into the gametes without influencing each other. They do not blend. They tend to separate and go into the different gametes without affecting each other. This is law of segregation, also called law of purity of gametes. Law of independent assortment, we talk about two characters, not one character. So out of the two characters, the alleles of each character segregate themselves in the gametes without influencing the other character's trait or allele. Understood? The statements are given in the description. Kindly go through it and learn it by heart. Laws of inheritance, very important. Now, the other question that is asked, none of the scientists are generally asked except for your Charles Darwin, Lamarck, and here we talk about father of genetics, that is Mendel, father of genetics. Now Mendel, Gregor Johann Mendel, please know the full name, Gregor Johann, not John, okay, please, and it's Mendel, not Mundell. Gregor Johann Mendel. Okay, this he was a pioneer for all kind of laws that we know about genetics right now. In the year 1884, we lost him, and then in 1906 it was found that whatever law and all he has stated, they are all apt, and then he was given a title of father of genetics. Now Gregor Johann Mendel worked on a pea plant. Please write the scientific name properly, children, otherwise no marks will be given if the names are not written in a proper manner. That is, the name has to be underlined and the first letter capitals. This letter has to be small and it has to go like this: Pisum sativum. Pisum sativum is the scientific name of the pea plant. Why pea plant was chosen? Because it has short lifespan, it had many characters which was visible, and many offsprings can be produced in one generation. So it's easy to study if you have many generations in a very short period of time. Imagine him studying genetics with human beings, maybe survival was an issue. So let it be. So Pisum sativum, very important. Gregor Johann Mendel, very important. And pea plant, very important. Characteristics of pea plant, very important. Dominant traits. Now there is a go in party. Okay, I know it is not making good sense in English, but it's very important to know the dominant traits. Okay, go is green. Okay, inflated, green and inflated, these are pods. Okay, then you have purple flower, you have axial flower, you have round seed, you have tall plant, and you have got yellow seed. Always remember, flower and seed is coming in party and pods are coming in go in. So green and inflated pods, then you have purple, axial, round, tall and yellow. These are all dominant traits, very important. Okay, they can ask you, the questions can be very important. Questions can be asked, identify the odd one out in which few can be dominant traits and one can be recessive. Remember, go in party. Go in party, green, and here yellow. Do not get confused with the color. Green pod is dominant, yellow seed is dominant. Now we have to talk about the recessive factors also. Green pod will have yellow pod as recessive trait. Inflated pod will have constricted pod, purple flower and white flower, axial flower and terminal flower, round seed and wrinkled seed, tall plant and dwarf plant, and yellow seed and green seed. Okay, this is go in party. Mark this important, go in party. Okay, after your boards. So this is the dominant trait. So when we have studied about this Mendel and his laws, there are three laws that Mendel has given: law of dominance, law of segregation, law of independent assortment. Statement is given in the description, kindly go through the same. Now next we move to the process, that is monohybrid cross and dihybrid cross. So the next topic, monohybrid cross. In the monohybrid cross, you can see that we have considered only one type of character. Now here I have taken the example of tall and dwarf. You can take any other example that we have just seen, the dominant traits and the recessive traits. Tall and dwarf, they have to be homozygous, that is pure line. Pure line means they are homozygous, that is they have both the alleles which are same. So homozygous tall, dwarf is a recessive trait, so it has to be homozygous. Now when it is a homozygous condition, you find only one type of gametes, and F1 will always be a heterozygous dominant trait. So heterozygous, here the trait I have taken as tall. Always remember children, in this case the gene is height of the plant. The gene is height of the plant, and the alleles, that is the alternative form of the genes, is being tall, that is capital T, or dwarf, that is small t. Okay, so you understood the difference between a gene and its allele. So here you have got heterozygous tall as the F1 plant. Now when F1 is selved, that is when F1 is crossed with another F1, you have the gamete formation as capital T and small t in each cases. So when they are selved, you have the formation of 4 possible combinations of the offsprings. In this case, you can see that the one is homozygous tall, the another 2 is heterozygous tall, and one is dwarf. Is that clear? So now when I have to talk about the phenotypic ratio and the genotypic ratio. First, what is the difference between a phenotype and a genotype? Genotype is the genetic composition of an organism or a complete set of genes present in an organism. Phenotype is the physical appearance, the observable traits in the organism is called the phenotype. The phenotypic ratio is 3 tall and 1 dwarf, 3:1, and the genotypic ratio is 1:2:1, that is 1 homozygous tall, 2 heterozygous tall, and 1 dwarf. You need to write and mention what is this 3 and 1 along with the ratio in the examination. Now please blindly know that for a monohybrid cross, the phenotypic ratio of the F2 generation is 3:1. For a monohybrid cross, the genotypic ratio of the F2 generation is 1:2:1. This number has to be by heart. Okay, so you understand the phenotypic ratio and genotypic ratio. Now in this case, the number is given in the simplest ratio. In your questions, the numbers can be given where you have to calculate the ratio and then come to conclusion. Like if suppose you are doing a cross and you have got 150 tall plant and 50 dwarf plant. So 150:50:3:1. So I have given you the phenotypic ratio. You can easily go back and calculate the F1, or you can calculate the parent. Parent are generally homozygous tall. If they have not mentioned F1 and F2 in the question, then the parent will be the just parent of that cross. Is that clear? Let us move to the next cross, that is dihybrid cross. Now next topic is the dihybrid cross. Now dihybrid cross you have seen, I have already done the cross to save your time. Now here I have taken the parent as round seed and yellow seed. Round is wrinkled seed and green seed. So this is round and yellow, this is green and wrinkled. Now when the gametes, since they are homozygous, so they will have the similar gametes. I have written the gametes in the circle, and the F1 you have got as, you can see here, you have got R, capital R, capital Y, small I, that is a heterozygous condition for the dominant trait, that is round and yellow. So you have got the phenotype. In the F1 generation, we generally don't write the ratio because it is only one type of offspring that is formed, which shows the law of dominance. Now here, since there are two traits taken, this is explaining you law of independent assortment. Fine. Now here F2, F1 cross F1 will give you the gametes. This is the Punnett square to show the gamete formation. Question can be asked to draw a Punnett square to show the gamete formation. So now with this gametes, I have done the cross here. You are going to pause the video here, make the cross and see the offsprings. The symbols should have the same phenotype. Is that clear? So now you can see here that this is all homozygous. This is homozygous recessive. This is homozygous dominant. These three and these three are the recombinant traits. What do you mean by that? You have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 round and yellow. 9 round and yellow. 3 round and green. 3 wrinkled and green, wrinkled and yellow, and 1 you have wrinkled and green, that is recessive, dominant and the recombinant. So you have 9:3:3:1 phenotypic ratio for the dihybrid F2 generation. And genotypic ratio is generally complex, but it's very easy to remember. What is a dihybrid? What is a monohybrid genotypic ratio? That is 1:2:1. Okay, double it. 2:4:2. And then again write the same. 1:2:1. This is your genotypic ratio for the dihybrid cross. It's never asked in the exam. It's very complex according to your syllabus, but it's very easy to remember. 1:2:1, 2:4:2, and 1:2:1. Okay, total 16, because 16 combinations are possible, so total should be 16. So this is your dihybrid ratio. Is that clear? The cross can be given. I hope, just check your crosses whether you have got the same ratio or not, and just put it in the comment box if you have done it correctly. Write yes in the comment box if you have done it correctly. So this is your dihybrid cross. So let us move to the next topic. Now next topic is sex determination in human beings. Now you all know that male has got 44 chromosomes plus XY, okay, and the female has 44 chromosomes, that is the autosomes, and XX allosomes. So this is your allosome or autosome. Okay. Now when the cells of males and the females undergo meiosis, it is going to produce two types of gametes. Okay, it is going to, you know that sperm is being produced by the males. It is going to produce two types of gametes. One will be 22 plus X, and the other one will be 22 plus Y. Similarly, the female is going to produce a ovum, which is going to be 22 plus X, because she has got only 2 X chromosomes, so egg will be of same kind. Okay, so depending upon which sperm is going to fuse with the egg, the gender of child is determined. If you have 22 plus X fusing with the egg, that is 22 plus X, you get 44 plus XX. So you have the girl child produced. Similarly, if you have got 22 plus Y fusing with the 22 plus X, you get 44 plus XY. Y. So you have got the male child formed. So who determines the gender of the child? It is the father who is responsible for the gender of the child. The father cannot choose that what gender the child will be. Okay, that is all on chances. But who is determining the gender? It is the father who determines the gender of the child. Is that clear? So next we will talk about X-linked inheritance. So next topic is X-linked inheritance or sex-linked inheritance. Now when you talk about sex-linked inheritance, any trait which is present on X chromosome or Y chromosome, that is a trait present on allosomes, determines the disease, whether it will have all the traits and the characters which are present on the X chromosome is called X-linked inheritance, and all those traits which are inherited from Y chromosome is called Y-linked inheritance. Now, when you talk about the X-linked inheritance example is hemophilia, color blindness, and Y-linked inheritance, that is from father to son. And X-linked is crisscross inheritance from father to the daughter and mother to the son. Okay. Now, X-linked inheritance example is baldness, hair on the pinna, that is on the outer surface of the ear. Right. So, when you talk about the X-linked inheritance, if you see a condition, it is denoted with capital X because X is a chromosome and it has got a trait which is recessive trait. So we give a symbol superscript with the small letter. We don't write small x because here X denotes the chromosome, not the gene. Okay, so you have to denote it by X, that is X to the power C for color blindness, X to the power H for hemophilia, or you can just write X to the power naught, which denotes that the recessive trait is present on the X chromosome. In case of females, when there is no trait present, the female is considered to be normal. If she has got one X chromosome with the defect, she is a carrier, she will not suffer from the disease. And if she has got both the chromosomes with the defect, then only she will be diseased. Okay, but in case of males, XY is normal, and even if the X present in the male has the disorder, the male will show the disorder. Why? Because male have got only one X chromosome. So whatever gene present on that X chromosome will be expressed in the males, and this X chromosome the son inherits from the mother. Okay, so that is why it is called crisscross inheritance. Okay, so this is how it goes about. We'll see few crosses and then mutation, and then it's over. Okay, we'll see few cases, like for example case 1. Case 1, let us have normal male and the female. So XX and XY. Let's have the Punnett square. So you get the habit of writing in the Punnett square. So female and male. So you can see that all the children to this couple will be normal with no trait because parents had no trait. Okay, this is case 1. Case 2, if you talk about, let us take a normal female marries a colorblind male. Okay, in this case, the female is normal and the male has got color blindness. So you can see that the daughters inherit that color blindness wala X chromosome from the father, and the son gets a normal X from the mother. None of the children will show the disorder, but the daughters will be carrier. No one will be diseased, no one will have the effect, but the daughters becomes the carrier. Now case 3, the carrier daughters marry a normal man. In this case, you'll find the carrier daughter, okay, and the normal man. So the first daughter is normal, the second daughter is a carrier, the first son is normal, but one of the son is diseased. So though the parents are not showing the trait has appeared in the next generation. So this is going to be a colorblind son. Colorblind son. Okay, 50% of the son will be colorblind. 25% of all the children are colorblind. So you should know how to get the percentage, right? Then next case, case 4, the carrier daughter marries a colorblind man. The carrier daughter marries a colorblind man. You can see X, X naught, X naught and X. So the daughter, first daughter is a carrier, the second daughter is colorblind. The first son is normal, but the second son is colorblind. So 50% of the daughters are colorblind, 50% of son is colorblind, and 50% of all the offsprings is also colorblind. And similarly, if you just make it case 5 that the colorblind female marries a colorblind man, then obviously all the children are supposed to be colorblind. So this is your sex linked inheritance. Cross can be asked, probable offsprings can be asked, percentage can be asked. We'll have few questions after the session is over. Let me see how much you have scored in those questions. Now next topic is mutation. Now mutation is sudden change in the number of chromosome structure or shape that leads to any kind of abnormality, you can say, but that is not contributing to the variations in the population. Please understand the difference between mutation and variation. Variation is we all are different, but mutation in the gene can be problematic for the outcome of the gene. For example, sickle cell anemia. In sickle cell anemia, there is just a point mutation where one of the alphabet, that is nitrogenous base, is changed, and because of that the amino acid produced is changed and the protein is changed, which leads to the shape of the RBC to change from a spherical shape to sickle shape, and that leads to internal clotting and the symptoms of the disease. So this is all from the basic biology. Now next is some questions. See to it that how many questions you could answer. Thank you children and happy learning.